complexity. For example, crystals of granite lack complexity, and mixed polymers lack specificity. They fail as specified complexity. There is a profound difference between order and information. Order, such as that found in crystals, is simply a set of repeated patterns. For example, a set of letters ordered like the order of a crystal might read ACFD, 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 and so on in a repetitive but ordered pattern. Order actually works contrary to information because order breeds repetition, and repetition works contrary to information. You'll note that in an information-bearing sequence of letters, such as the cow jumped over the moon, we have specific placement of letters based on information. However, there is no order. In fact, if you were not able to understand the information in the sequence, you would find absolutely no rational sequencing in the order of letters whatsoever. From here we'll take a specific look at an example of specified complexity in biology that was brought up in the video. The first would be that of the optical purity of amino acids, and the second that of the information or assembling of DNA. And then we'll let two competing hypotheses, one of law-constrained chance, chaos theory, and that of design, function, and see whether chaos theory actually takes the evidence and has any explanatory power beyond that of design. In order to understand the weight of the argument of the optical purity of amino acids as a design argument, it's first important to understand the nature of an isomer. Isomers are simply chemically identical mirror images. This first molecule is built with what we'll call atoms A, B, C, and D. Its isomer is also built with the atoms A, B, C, and D. Notice here how the correspondence between the atoms and the relationship between them is the same in each isomer. A is next to B, corresponds to C, corresponds to D, all corresponding to the carbon atom the same way, except they lay reversed, and therefore they can't superimpose one another. Now what's important to understand is that nothing in natural chemistry can identify or recognize the difference between isomers. Also, whenever isomers are formed in nature, they're formed in what is called a racemic mixture. 50% of one type of the isomer, 50% of the other type of the isomer. Now, depending on which way the atoms rotate, we label these either L isomers or D isomers. So we'll call these L amino acids and D amino acids. Now, in life, only L amino acids are used, and Ds are toxic to life. So the question arises, if nature produces 50% of L's and D's together at all times, and yet life was formed with a collection of just the L amino acids, what occurred in order for the L's to be separated from the D's so that D's not only are not used in life, but are actually toxic to the information found in life? As an illustration of how isomers work, consider two puzzles which are cut from the identically same die, so that the corresponding pieces from both puzzles are shaped identically the same, but the pictures on them are different. Now the illustration here is that the puzzle pieces are isomers, and that the pictures on them are the differences in the rotation of the atoms. But nature is a blind puzzle builder, so it would be the same as taking the puzzles, breaking them into their respective pieces and mixing the two puzzles together, so that we'd have a racemic mixture of the two puzzles, like a racemic mixture of isomers. But then rebuilding the puzzles blindfolded, so that you would be blind to the actual differences, which would be the pictures on the puzzles. You could structurally put the puzzles together, but you'd never be able to recognize that there was a wrong and right piece for each location in each puzzle. So you'd build the puzzle with both pictures in it. In the same way, nature, being blind to the difference between isomers, would build the puzzles of isomers using both L and D forms. So we would expect life to have both the L and D forms of isomers, and yet the puzzles in nature have been made using just the L forms. So in other words, nature is a blind puzzle builder and yet contains perfect puzzles. The most popular naturalist theory argues that the answers to this evolutionary conundrum are found in space. This theory is offered because we know that circularly polarized ultraviolet light is known to be able to decompose selectively either the L or D forms of amino acids. We also know that light from stars, as they pass through charged particles of dust, can be circularly polarized. So the theory states that amino acids, either traveling through space or while on Earth, were bathed with circularly polarized ultraviolet light, which then created an excess of one or the other isomer. In this case, we'd say an excess of L isomers. Life then began on Earth with a disproportionate amount of L's to D's. Then the theory states that some sort of auto-multiplication process occurred in which life automatically purified itself so that it continued to replicate itself, removing D amino acids until life had only L amino acids formed. This argument is based on some research which was done at the University of Tokyo on the autoreplication and purification of isomeric molecules. Now, researchers at Tokyo found one isolated molecule in nature which has the ability, when created with a small enantiomeric excess, to auto-replicate and purify itself to about a 99% purity of the isomer which was in excess. So the theories of biological autopurification are based upon this research as foundational evidence. However, there are many reasons why the University of Tokyo research is foundationally irrelevant to the optical purity of amino acids. First, the only biological relevance of the experiments at Tokyo is that an amino acid is used as a catalyst in the process. The actual subject molecule which auto-purified is pyrimidyl alcohol. Second, the results with this pyrimidyl alcohol have been limited to pyrimidine 5-carbaldehyde, a biologically non-useful carbon atom. And third, the actual process of autopurification is a laboratory-induced result that requires purposeful stimulus with particular specifications. As the parameters of these particular specifications narrow, the amount of complexity and specification increases. Thus, rather than provide a working, natural explanation, it only increases the markers of specified complexity. Other theories or other problems which confront the space theory, again, is that the circularly polarized light can only create a small enantiomeric excess. So again, what we have on Earth is a pure life form, pure amino acids of one type. Other theories that are offered is that life began racemically, with both L and D amino acids in it, and a process on Earth purified it. But again, the theory is based in sole conjecture. It's a theory which is trying to argue naturalism because of the presupposition of naturalism. It's ignoring all evidence which states that there is nothing in nature which can do the purification process in an amino acid. So what we have then in the complexity of amino acids is a very specific complexity, a complexity which points to life, which is necessary for life, which is unexplained by constrained law. Although some try to explain it, again, we'll look at chaos theory in just a moment. It's unexplained by constrained law, and yet exists and provides a specific purpose. So we have a specified complexity, and I would argue that the specified complexity then is a clear, actual marker of design. The next step we would take from the optical period of amino acids is the information actually found in DNA. Let's discuss momentarily first what information is. How do we recognize information? Information, first of all, isn't found in constituents. Information is found in the arrangement of constituents. You may think of an alphabet, or the letters T, C, and A. T, C, and A alone, the constituents provide no information, but in the order CAT, they do provide information, cat, or feline. So we understand that information is contained in the sequence-specific arrangement of constituents. Second, information is a non-material entity. For example, think of a floppy disk. If I held a floppy disk and it was empty, there was no information on it, and I weighed it, and then took it and filled it to the maximum with information from my computer and weighed it again, the weight difference would be negligible. There'd be no change.